Okay, I'm going to tell a little bit about metabolism. Very simple models I've been doing with many people here, and just to, since I'm going to run out of time, why is not moving? Oh, it's moving. I just want to point out that Donia G was the main guy doing this work. Herb has been involved, so is Jason George, another professor that was a graduate student center, and two experimental collaborators, mostly Benny Captru and Vito Cantana. So I just want to point out to remember that the people have been involved on this effort. So this idea starts on a very simple idea that basically, as everybody knows, I'm going to go that based during cancer, you have aerobic glycolysis. That's something you don't have in regular situation that you have. So it started to work on this system just for fun. And actually, there are interesting things. Okay, it's just moving slow. There are interesting things I want to point on this plot that basically, if you look metastasis going around, we start to observe the most aggressive cells. They actually use all the resources. They use not only glycolysis, but they also use phosphorylation. They try to make all the energy that's possible. And if you look, for example, in terms of gene expressions, you see that basically the ones that actually are in the circular tumor cells are the ones that actually have the largest expression of these things as they are moving around. So we decided to go after this problem many times. Basically, you heard a little bit. I talk about hybrid states, and that's the metabolism version of the hybrid states. We start doing these things in terms of epithelial-mesenchymal transition, but you're going to see that these hybrid states, where you have this dual performance, they also get coupled in all the systems, basically. And if I have time, I may try to get this situation. So I want to say that just for historical reasons, this is an effort that started when we went to Houston, and Eshel Ben Jacob was involved on it. He was a main player here. And I will see in the beginning of the story, there's a lot of things going. I have doing some of it now. Herb is doing a lot of it. But Eshel was actually part of this team when these things started. And I want to just point this for the fun here. And now we're going to do simplification, just to get people here talk about complicated systems and make it a lot of fun and make the system much larger. We're going to say, how about if we throw out most of everything and just keep a few super genes and try to work on the system. So in a certain way, everybody has been looking on the systems on metabolism, and this is like a simplified version already of a very complicated version that people draw for you. But if you have a look, you start to observe there's some master regulators like AMK and HIF1 here. They appear to be connected to everything, and they appear to be controlling everything. So with this, you try to play a game that says, let's focus on this system as this and see if I can draw simplified circuits. Drives many people crazy by saying how you're sure you have everything you need. They're going to see how you go after the experiments and how the comparison is done, just for some entertainment as building these models. So let's try to move forward on that. So the simplest circuit when you first start was just a circuit like that. So what's very nice about that circuit is that basically, and here, the data I'm going to present most here is just deterministic solution of these things. You can actually do putting noise, and as you add noise to the series stochasticity, these features I'm going to show here, they actually get amplified. But with a deterministic system, you can do very simple equations and already see how the system behaves. So what you observe on these circuits, and we're going to do, and I'm going to present to you, is the following thing. If you solve this system with regular parameters that you have on healthy cells, you're going to end up, when you put the two nucleons, you're going to end up just with states, the ox state or the glycolysis states. However, as you start to push the system, you put the system under stress, and here can be includes mitochondrial loss, can be stabilization of HIF1, a few other things that are there. What you're going to see is that the system creates this hybrid state under stress. The same way you observed on epithelial-mesenchymal, you can solve the system, and you'll start to observe that you have this state that's a mix of the states of, that's a mixture of the glycolysis and the oxphos state. Anyway, that's not for you guys, so I'm going to jump this slide. This to show that basically all you're solving are equations of this form, a little bit more complicated, drawing nucleons and going forward. But what's very nice is that these things at least agree. I want to stop a little bit on this slide and tell you about situations. So, for example, you cannot look, for example, just at the expression of HIF1 AMK because they don't give you enough information, particularly AMK, because the phosphorylate one that's important. So if you just look production of AMK is not going to help. So what you have to see is to look at downstream genes to do that, and then do the comparison of these downstream genes. And you actually see that this appears to be very good for at least qualitatively the model you're trying to say. You have this really anti-correlation. Here's patient data, about hundreds of patients in each one of these, and then there's single cell data there. They all show this anti-correlation as we expect on the previous slide. Not this one. If you expect, basically, in the fact that you have this toggle switch, basically, you're controlling these things here. So that gave us an idea that basically this is not a bad way of going after this system. But as I said, this system is a little bit too simplified. So we decided, okay, can now we couple this genetic network to the entire metabolic flux, right? Basically, can you couple the two things? So we generalized the circuit a little bit more. And I think now here's the simplified version of where you have base AFK controlling both glycolysis and OXPHOS and coupled to metabolism. The cool thing about it is that now we can explore these models using, let's assume this topology of the network is sufficient, and you can explore the entire parameter space of it. So you have a technique called recipe, where you pretty much build many times these models with parameters where you try to be physiological, but you move the parameters around a lot. So you do like 1,000 different parameters, and we compare what's happening here. So what you observe here that's kind of cool is that now, as you explore about 1,000 possibilities on these things, and you do this sort of gene expression, and you organize this in terms of genes, you start to see that you actually see these three states appearing with minor fluctuation of these parameters. They cannot be just physiological. They have to go to the stress regime. Otherwise, you don't see this state appearing. What's kind of cool is that basically, if you go to the patient data here, you have to be a little bit more optimistic, right? But you start to see the same behavior that if this simple model can give. And I think I have a better representation here that tells you you can find basically something that looks like your OXPHOS. You can find your glycolysis states, and you can have something where you see gene expression of both regions, the region that is OXPHOS, that's a hybrid state coming together. So this is a powerful of these models because tells you that basically the topology of these networks are very robust. What's changing here is these parameters telling you that basically as the system is going to stress, you're pretty much changing all these parameters and you're going to go this situation. Now, it's kind of cool because if you go back to the data, and you look here, your anti-correlation, you see exactly that basically these states, you look at this patient data, you see exactly that you fall on the basically OXPHOS hybrid states, and the ones that this anti-correlation is there when both groups are working. Now, Benny, who actually inspired us, a great experimentalist, look at the system, he started looking at this data and say basically, that makes sense. And you observe now on the laboratory, I'm not going to go all the details, we start to observe here that you have a state here that you put like an OXPHOS inhibitor, you go from OXPHOS state to the glycolytic state. It will go the other way around. If you look, for example, here you're showing both OCR and ICAR, so you can follow both OX4 and glycolysis. You can see here basically if you put now a glycolysis inhibitor, we start to observe that basically the PMK goes up, but not the AMK, what tells you why you have to follow the genes down forward. Here is some genes forward, but I want to just, in the interest of time, show this slide is actually the conclusion that you have on that. That basically, if you just do these cells, these aggressive cancers, and you attack them with some glycolysis inhibitor, doesn't do it too much. If you put just OXPHOS inhibitor, doesn't do you too much. You actually have to add both of them to go together. Basically, here's just a comparison. Even here is cell proliferation, but because in the percentage of colonies get even, these doubts get more dark. If you like, I love this slide here that you really see that basically only when you add both glycolysis and OXPHOS inhibition, the system goes down. Now, Herbie was never happy with that, so I'm going to blame him for that. And then he started to look for people around that could generalize. And the idea was the following, basically. Why is the system breaks the symmetry? And if the cancer has high, low, low, high, 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 why not low, low? Why cannot have a system that basically you pretty much put both of them down? And actually, that's where Vitor Quintana and his collaborators down in Tennessee came together with us, is basically, if you really push the system hard, and that's pretty much increasing, like, for example, mitochondria ROS by large amounts, you can add some states that you're missing, and you find this low, low state. Now, the experiments we have, I think they support this idea. You're going to tell me that it is a funny story, basically. It's here. They have these three cell cancer cell lines, and basically they use BRAF1 that basically inhibit the systems. And I can go to overall these plots, but I want to show that if you just look this one down, you pretty much start to a state where you have cells with both signature of both of them and comes down. All the other plots support this picture. We don't have enough time to go in detail to all of them, but it's kind of cool to see that you go up to this low, low state. It's not a state that you pretty much stop growth. It's almost like the system stay stable. It's like you grow at the same time you die, so it becomes just like these very boring states. But these are very tough states because the states that don't respond too much, they're the most things that's very hard for you to put any therapy to go there. So that's a sort of a cool story that basically came from the theory. We search for the experiments. You bring these things together, and the system now give you an idea of how these things are coming together. Sorry, I'm going too fast, but it's on these 20 minutes. I'm trying to cover a little bit just to get a feeling, but these are sort of general ideas. So what I want to stop here is to say that basically this is a different way of looking at these things, is actually oversimplify the models and see how far can you go with it. I think there's two ways you can do it in this situation, right? And then basically say, what are we missing? So there was a lot of things we're missing. And the experimentalists start complaining about us because there's this control by MYC, that's something we're working big time. We haven't dealt with glutamine properly. So he sort of, Donny Adjia came at, he made like another improved, Jason George started to work with that on that model. And what's cool is that basically you can generalize this model and see now what gives to you. And actually, you can see that now you have when you look here, the nucleons and see which states you have, you start to observe that now we get 4 states, and you have this Q state here that basically used to be our low, low states, but now we just changed the name, what we call glutamine-dependent states. It's a state where glutamine becomes high. So it's nice to see that it's kind of cool, something that comes from the theory. You predict you need something else, you generalize the model a little bit, and you get this picture in front of you. The other thing here is on the generalized model, you can actually see the anabolic and catabolic process. Everything starts to gear, and you have a few ideas of how the system should go. And if you want to go after some drug therapies, you can go after that. And that's basically what some of the experiments we're doing these days are doing now. Is very interesting to observe. Now, with these states, you can start to look at patient data. And this is sort of I look at this thing and say, do I explain anything? No, I don't explain anything, but these are very nice observations. It's basically why when you have a liver hepatopoietic cellular carcinoma, these sort of low, low states tend to be the one with worse prognostic of life extension, but on this, by their case beers where the hybrid states is the one. So it becomes very interesting to observe. This is just observation. It's collateral adenoma carcinoma. Why these two different cancers have the cells that are more, which patients tend to have worse prognosis is not, but it's an interesting thing to look around, right? So you can say that basically, and these are things that actually are cool that you have with this very simplified representation. So now there is the question, like I said, we told you guys before that basically all of these cancers, you have to have both these inhibitors for glycolysis and inhibitors for OXPHOS to have your system to behave. And actually, basically, these are lots of time-dependent experiment situations that we are getting here. And you start to observe that basically you can move these states, and then now you can start to simulate the system with different ways based how you turn on, what you turn on. Basically, particularly, this was a big influence from Escher. Escher loved metaboforming. And so the question is how it works. And you can see that basically you have these things with dual drugs that do that. But there's a very interesting result of metformin, because I think I'm getting close on time. I want to show that comes on the next slide. That is the following. Metformin has this very funny situation that can affect directly into the ATC or can modulate AMK. So what you observe on that's a sort of very nice result, and you can see from the model, is that if you just put a little bit of metformin, you actually, because of the AMK, you accelerate the process. So actually you have to go to higher concentration to affect the ATC for the system to come down. So this becomes a very interesting thing that we are trying to work these days, is the fact that basically not only these drugs affect, but the concentration of drugs becomes very important. I think the experimental data is not as radical as the simulation we have up there, but you see this behavior. And actually, there are a lot of people giving cancer patients drugs on this regime, and they say no effect. And basically here you have to see that basically you have to at least, you don't want to give too much to kill someone, but you have at least to cross this threshold to actually start to have any therapeutic effect as you move into the systems. I think I'm running out of time, and I was going to tell you a little bit about the couple between metabolism and EMT. But what I want to point out is the following. These systems are all coupled, right? And basically, and metastasis is also associated to a hybrid state of this one. But what's cool is the following. You can have systems where if you do just a study of the circuits, EMT circuit or metabolism circuit, they are okay. But if you couple them, you can see that if you push the metabolism to go hybrid, it may drive EMT to go hybrid. Or the other way, if you put EMT to be hybrid, you may drive they start to go. So I'm going to stop here because I want to stay on I'm looking my time here and my 20 minutes.
You still have a minute or two, but you can use it for questions now.
Okay, and that's good. It's a good place to stop.
Thankful.
Well, going.
I have a question, but maybe someone else first, but other than that, I have a question. Okay, so the previous slide.
Which one?
22. One more one previous here. So I noticed that in a lot of your things, you sort of have these non-monotonic responses that keep showing up. I was wondering if one can look at these low-dimensional outputs, like these things here, and say something about what might be happening in this.
No, here actually you see, because basically this is a clear case that basically lower doses of metformin attest to affect this control here, when higher doses affect the OXPHOS down here. So, right, it's a question that basically metformin control both of them, but the level they control changes on what they do. So, and that's where it gives this behavior.
Okay. Any other questions? No? Okay, Pankaj.
I have a general philosophical question. How much do I need to know about the system before I can really start writing models like this? Like how much?
Okay, I will tell you.
It's a very honest question, right?
No, it's a very honest question.
And how?
Okay, I will tell you that this applies to almost everything I have done in my life. It's like when we did protein folding, and now when we're doing genomic models and things like that. The question is, how much data do we need to start to do it, right? And the trouble is, is like physicists somehow they have some aversion to data, right? They want to say, what's the minimal things I need? So it is a situation that basically Escher was very good on that, on the thing that he moved on that. I'm much better on the molecular level. But the question is, you start to read all these papers and you try to create on your mind, try to build a picture. Now, the dangers of that is that it's very easy to get in love with your own ideas, right? So basically, you may believe you have a model. That's why I think, in a certain way, you have to have this sort of experimental proof of the pudding into the end. And that's why I think all these collaborations that we have in every system we have, the same way we have here, we have on chromosomes, we have some experimental partners that actually are supposed to keep us honest, or you're supposed to keep them honest. You can see it on this way. But that's a very, very good, it is a very good question, particularly try to come to some very small models like that. So that's a philosophy that basically, and as you can see, as we move through the experiments, and I went a little bit too fast here to make the story, you see that we start in a very simplified version, and we start to complicate as we felt that we could accommodate everything, right? And then basically these models start to grow a little bit, and now they're on a level that we believe we can accommodate all this drugging situation, but it's a question to see how well this is going to work all the way.
Okay, there are no further questions for now. Let's all thank Jose again.